All right, welcome everybody. We are going to go ahead and get started with our NBIA research overview. So if I could please have everyone just get seated. We'll get started in just a minute. As you know, Dr. Susan Hayflick was unfortunately not able to be here this weekend due to a family emergency. And so what we are instead going to do is have all of the presenters who gave updates on the individual NBIA disorder research updates just now come up and give a very short condensed version of about 5 minutes of what they said to their individual groups, so that way all the disorders can get a sense of what is going on in the other sessions that they were unable to attend. And I am going to start with PCAN. Can I have Dr. Richard Lee or Dr. Ben Mamoun come up? Thank you. And so it's kind of going to be a lightning round where everyone's just going to get 5 minutes to kind of tell the whole group what they're working on their disorder. Thank you all.
Oh, it's a big audience. I'm Richard Lee. I'm from St. Jude. I'm a medicinal chemist by training, and I'm leading the PCAN discovery group at St. Jude now after Susan Jukowski retired. What we're doing is we're trying to make pancake activators to treat PCAN, and we're following up on our previous compound series, which ultimately produced the BridgeBio lead compound BBP-671. What we're doing is we're taking our lessons we learned in that program, and we are going to try and eliminate the problems we had with BBP-671, which was a remarkably successful program in that we were able to get a compound into the clinical trials, into the clinic and prove it was safe. The problem with the compound was that it was metabolized a little bit too quickly and it wasn't well absorbed in the gut. So we were thinking about the problem was getting it into the brain, and the brain was going to be the big problem. But it turned out actually it was absorption through the gut that turned out to be the issue. But anyway, we've just not restarted, but we've refocused the program. We have a number of new members of faculty at St. Jude and some new scientists and some new approaches. And we have this beautiful data package from the development of BBP-671, and we're moving forward. And hopefully we'll be able to get a new compound, a new clinical compound very soon. And thank you.
Much shorter. All right. Hi, everyone. I'm Jessica Regan, and I am the Scientific Director of Virtus Therapeutics. We're a spin-out of Dr. Shukri Ben Mamoun's lab at Yale University. And we're working also on pancake activators for the treatment of PCAN. So our program, we have identified a series of different compounds that activate pancake-3 with effective concentrations in the nanomolar range, which is really low concentrations. These compounds activate the enzyme itself, but they also are able in patient cell lines and in pancake-2 deficient cell lines, which mimic PCAN patients. They're able to increase coenzyme A activity, which is important for the overall underlying process of this disease. These compounds are able to cross the blood-brain barrier, which is really important for a neurological disorder. So we're really excited about the chemical properties of these compounds. We then moved into animal studies for these compounds, and using a whole body knockout model from OHSU that they reported on, we were able to identify that our compounds increase coenzyme A concentration in the brain of both wild type and pancake-2 knockout mice. We then moved into another model that was previously reported by St. Jude. We found some really interesting features of this model that unfortunately had not been previously reported. However, that being said, we're working on expanding those studies, and we have some promising preliminary data with those animals, and we're really hoping to have some more exciting data within the next 6 months to a year for that overall project. So we're really excited where we're at right now, and we're also kind of leaning towards an additional route where we're looking at biomarkers, since there's currently no available biomarkers for PCAN or other NBIA-related disorders. So we're really focused in, and we identified a top biomarker among 49 additional backup biomarkers that seem to be upregulated in patient cell lines, as well as PANC2 deficient knockout cell lines that we have engineered in the lab itself. So we're really excited about this, and what's really exciting for everybody in this room as a whole is we found that this biomarker might not only be important for PCAN, but it might be important for other coenzyme A disorders in addition. So we're really excited overall, and that's where we're at. So thank you.
Okay, we'll now turn to our lightning round for BPAN, and we'll start with Dr. Ayuso, please.
Hi everyone, this is Arcangela Ayuso. I come from the Helmut Zentrum in Munich, Germany, and my research focus has been investigating the role of mitochondrial dysfunction in the pathogenesis of BPAN. So we have investigated so far fibroblasts from different patients, and we saw that mitochondrial dysfunction is quite a common feature in BPAN. And not only fibroblasts are affected, but also brain cells, such as astrocytes. We have also initiated a sort of a therapeutic treatment to ameliorate mitochondrial function, stimulating the cyclic AMP pathway. So far, we have tested 3 different molecules, which are giving a promising result. Surprisingly, dopaminergic neurons are not affected. We still don't know why this is, but we have some ideas, and we want to follow up to understand better this aspect. And this is where we are. Thank you.
Morning, everyone. I'm Dr. Joe Vitheathel from Children's Hospital of Philadelphia, and I'm one of the providers in our BPAN clinic there. And so we are conducting a natural history study at the Children's Hospital of Philadelphia in our clinic, and our update was just sort of on some of the outcome measures that we're identifying in the plan to be ready for clinical trials. And so the big issue is making sure we have the correct outcome measures and assessment tools that we want to use during clinical trials. And so we've identified a couple of the validated outcome measures that are used predominantly in the cerebral palsy community, but have been very effective in being able to assess our patients with BPAN. So we'll be continuing to validate those with larger cohorts, and then we're starting to gear up to hopefully develop more treatments in the space of gene therapy to be able to use those in clinical trials. Thank you.
Next up, we'll have some updates on PLAN, and it looks like we'll have Amber Gregory and Dr. Jenny Wilson from OHSU presenting.
Just told Dr. Wilson this feels like the Oscars where you have to do the long walk and you might trip. So first of all, again, Susan Hayflick would love to be here. I'm so sorry she's not. I think that, in 2 years we'll have a very happy reunion, and she can tell you a lot more about what's going on with our group. I'm going to just give you the 2-minute overview of everything, because I think that covers a lot of people in the room, but we do have a lot of balls in the air. You all know that. We still have the natural history studies going for BPAN, PLAN, and PCAN. And I just presented in another room some of the PLAN data. We got a little grant to do an analysis just for the INAD subgroup, which was about 40 people, and that's going to be published in the near future in Developmental Medicine and Child Neurology Journal. So that's exciting. It'll help lay the foundation again for clinical trials. During the PCAN clinical session, we gave a quick update on our CoAZ product, but I know that's of interest to everybody. So this is a product that was in the work for about 10 years now, and it feeds into the CoA pathway sort of just past the block. It's like a traffic jam with the enzyme that doesn't work, so you can't push things through the cycle to get to the end point of CoA. Ooh, sorry. And we've analyzed our data from the first 6 months of the trial for CoAZ. That was the double-blind period where we had a control group and everything was done very rigorously. And that data has really nicely demonstrated a dose-response relationship where the low dose of CoAZ showed a change and impact on our biomarker, and a medium dose showed a greater change, and the high dose, it all tracked, which our statistician who is usually sort of a very calm person, was very excited, which is a good sign. So we submitted our food additive petition to the FDA in February to try to have that product approved, not as a drug, but as a medical food, which means that you wouldn't need a prescription to get it, that you still need to be supervised by a doctor while you're using it, but it's a pathway that we chose that's separate from a drug pathway through the FDA. So all that is to be continued. It is in the works. And then Dr. Wilson and I will give you a brief update on the pecan gene therapy that we're also working on. We're wrapping up. So we've been doing, for the last year and a half, 2 years, it's been like all mice all day and night around our place, a lot of pecan mice, doing studies to choose the best design, the best what's called a promoter, the thing that sort of drives the gene, like turns it up or down. Looking at the durability of the effect, that's what we're finishing up right now. We've chosen like our #1 vector and the promoter and everything, but we want to make sure that we continue to see the effect of putting that gene in a mouse after 1 month, after 6 months, after 9 months to prove that it will have a long-term effect. And then Dr. Wilson's been working more on what this is going to look like for humans when there's a trial.
Yeah, so we're just kind of thinking a number of steps ahead. We're still in the mouse phase. And this gene therapy would be delivered through injections directly to the basal ganglia, the deep structures in the brain that are affected in pecan, the eye of the tiger sign. So it's given in a different way than other gene therapies. Other gene therapies may be given through CSF, sometimes blood, but this would actually be a direct injection, a one-time injection. So we'll likely start with an adult with pecan as our first patient, but the FDA is open to us moving to children pretty soon after that, and we'll likely have a handful, maybe 5 to 10 patients early on in the study where each patient is separated by a few months, so we're carefully looking for any adverse events after each patient before moving on to the next. But still some time to go on that as we're still working with the animals.
And I think last we have Dr. DeMaio. Is Dr. DeMaio here? Yes. Thank you.
Good morning. My name is Ivano Di Meo. I come from Italy, Milan, the Institute of Neurology Carlo Besta. Our group is involved in different projects on NBIA, different form of NBIA. Today I'm here because to present my project on PILAN, which is called PILAN Mark, which the aim is to find a circulating biomarker for PILAN to help therapeutic readiness. So this project is started at the end of last year, so it is designed spanning 2 years. During the first year we are collecting all the samples, enrolling patients and collecting samples, circulating samples such as blood, plasma and CSF. And once we have all the samples, we will move in laboratory to analyze all these samples in different levels, transcriptomic analysis to analyze all the genes expressed in the blood to find differences in patients and healthy controls, to find a molecular signature of the disease of the patients. The second level is the analysis of circulating microRNA, which are small non-coding RNA, which are actively secreted by cells in the body fluids and have been associated to differences in microRNA expression are associated to different kind of neurological disorders. So our aim is to find potential microRNA differentially expressed in patients. The third level is the targeted proteomics, so using a new platform which is called NuLISA, which is able to simultaneously measure 120 proteins which have been proved to be associated with the neurodegenerative and neuroinflammatory disorders in order to find a possible protein working as a biomarker. And third level is the measure of circulating cell-free mitochondrial DNA, which has been demonstrated to be to accumulate in body fluids after mitochondrial injury or oxidative stress or neuroinflammation, which are typical of PLAN. And also, we are also working on other type of NBIA. For example, we recently generated some different type of mouse model for Copan, which is a very rare form of NBIA. And now we are testing some compounds in these mice. And also, we are developing, just started to develop a gene therapy approach based on the AAV vectors. And also we have some IPS derived from PCAN patients and also Copan patients. So we are also working in study the pathomechanisms and the potential therapeutic approach for PCAN and Copan, linked to coenzyme A deficiency. And thank you.